Okay, so I'm going to be explaining DNA replication by, you guessed it, the zipper model. So here we have the 5' and the 3' end of the strands, and the zipper here represents helicase during the unwinding of the two strands of DNA. As the strands unwind, single-stranded binding proteins, represented by these little push pins literally holding the jacket together, prevent DNA from zipping back together. DNA replication is going to the left here. The leading strand is the one with the 5', where the continuous replication can occur. But the 3' end is where discontinuous replication occurs. It's the lagging strand. That's because DNA can only add from 3' to 5', so it's going to add in small chunks, like this. These small fragments are called Okazaki fragments. To relieve the tension from the unzipped double helix, gyrase, represented by this T bag, relieves the tension and prevents supercoiling. Get it? T, it relaxes you. Next we have DNA primase, which lays down RNA primers for the DNA polymerase to bind to. And the primers are represented by M&Ms for the DNA polymerase to bind to, because who wouldn't want to bind to M&Ms? So DNA polymerase 3 is actually going to continuously grab this new strand and add nucleotides to this strand here. Same with the lagging strand here, the DNA polymerase 3 is also going to add nucleotides in those little segments on this lagging strand from the 3' to 5' end. Then, to continue the lagging strand, the DNA polymerase run is going to remove these primers so that the DNA polymerase 3 can continue to create the lagging strand. This actually creates nicks in the DNA strand. So ligase, which we'll be using as a band-aid, will cover these nicks so DNA polymerase 3 can continue to add to the nucleotide chain. DNA polymerase run is actually very special in that it has an exonuclease activity that can edit and proofread from 5' to 3', and if it didn't, it would only edit from 3' to 5', and it can only remove one error at a time. So if it couldn't do this, then we would have a lot more errors in the DNA and it wouldn't be able to replicate successfully. Thank you for watching my representation of DNA replication for prokaryotes. All of these kind of silly little supplies are mine, but the information that I got for this video was Module 4, Video 4B for replication, as well as the Raven textbook.